Hello, Anita Miss. Shall we start?
Yeah, Divya, over to you.
Okay, fine. Hari Om. A very good morning and hearty welcome to all for the 17th webinar series hosted by PG Department of Biotechnology in association with IQAC, Chinarts and Science College for Women. Today we have a session on how drugs are developed, biopharmaceutical application of scientific research by Dr. Sanil Sansar. As we all know, drug discovery and development is a very expensive process, both in terms of time and money that is being invested. Now, it takes a lot of more than 10 years for any drug to reach the market. And if a drug fails at the final stage and all means, it incur a huge losses. So commercialization of any drug is a tedious process actually. So today let's listen to Dr. Sanil Sansar in detail about this journey of a medicine from lab to shelf. On behalf of Chinmay Arts and Science College for Women, I bid a warm welcome to our resource person, our principal who will join us shortly, other delegates and all the participants who have joined us today. I guarantee that the session would be quite productive and obviously worth your precious time. May I now invite Head Ms. Naina Sardevan, PG Department of Biotechnology, to introduce our resource person to you all. Over to you, Naina.
Thank you, Divya ma'am. Hari Om. I'm so elated today as our PG Department of Biotechnology joins hands with the IQAC of our college, headed by Ms. Anita Haridas, to witness the international edition of our Chinarts webinar. I'm indeed privileged to introduce our resource person of the day, Dr. Sanil Sansar, to all of you. Dr. Sanil Sansar is a mission-driven medical research professional with proven experience in all aspects of stakeholder engagement, clinical trial coordination, and development. Currently, he is serving as a medical science liaison at the Kadmon Pharmaceuticals, Dallas. Earlier, he played several crucial roles, such as a clinical research liaison at MedPace, Dallas, as clinical research coordinator at Texas Scottish Rite Hospital for Children, Dallas, as postdoctoral research associate at Texas Women's University, Denton, and as a teaching and research assistant at the same university. His core competencies include hematology, oncology, rare diseases expertise, medical affairs stakeholder relationship management, product lifecycle management, scientific translational research experience, and teaching and scientific presentation. He was awarded a PhD by the Texas Women's University, Denton in 2017 for his work in neurobiology and molecular biology. He has given research presentations at several prestigious symposiums and is the recipient of many awards and honors for his excellence in academics and research. It is a matter of pride to have such a phenomenal personality amongst us. On behalf of Chinarts, let me extend the warmest welcome to you, sir. Divya ma'am, over to you.
Okay, thank you, Naina. So before we start, participants, please note a few instructions. Kindly keep your mic muted. You can post your queries in the chat box towards the end of the session. As we know, we are live streaming on YouTube channel as well, so you can post your queries there also. We'll be having a QA at the end of the session, and the attendance form and feedback form will be posted towards the end. And we also have a recorded version of the webinar, which will be available later on our YouTube channel. So that's it. So in this virtual conference room, I wish you all a very interesting and successful webinar. Now, without further ado, let's turn the time over to Dr. Sanil Sansar. Over to you, sir.
Thank you very much, Divya, Divya madam, Naina madam, Jisha madam, and Principal Dr. Lalita. Thank you once again for the very kind introduction, Naina madam. It has been very kind, and it was a pleasure to hear that, I had long forgotten many of those. My dear students, I would like to set the expectations before I start. What I would love to do is I would like to keep this floor or this session very informal as possible, which which I mean that you could be asking questions anytime. You could stop me if I'm not clear for you, if I'm not making things clear, if you need further explanation. I'm okay with taking questions anytime during the presentation or after the presentation. It looks like usually you do the questions at the end of the session, but I'm fine with any of those. To make the best use of this time that is given to me, I would like to make a few things more clear. What I would like to do here is the intention here is to give a brief idea about how drugs are developed and help you understand there is a world, an entirely different world out there other than academic research, which is like the postdoc, PhD and postdoc, master's, PhD, postdoc, the regular route that you usually take. And it is called the industrial research and pharmaceutical research. I am doing this. I took up this topic and I'm doing this because when I was growing up and I was going to college and universities and then doing my PhD, I did not have any resource person to guide me. When I was interested in going into the industry, I never got a guidance for myself. And since I have gained it through hard, hard research and hard work, I would like to impart this knowledge to whoever is in the receiving end and whoever would benefit from this. So to take maximum advantage of that, please feel free to ask questions regarding any of those. Let us jump right in. But I would like to, it is really, I don't know if you have ever presented to a big team like this. I have, but the difficulty in this world right now is that I'm talking to a computer monitor that I see 365 days. I'm looking at the same monitor I look at to work as well. I usually love to see faces so that I know how you receive it, so I can reciprocate better and gauge what your understanding is and then give more or less depending on whatever is being received. And I would really appreciate if I can see faces. If not, that is fine too. So let us jump right in. So the topic right now is, like Divya madam mentioned, it is a difficult task to develop drugs, and how difficult it is we will find out from this title page. You could see that about when you test about 1,000, 10,000, thousands and thousands, like 10,000 compounds, what you end up is one compound being approved after all the clinical trials and research and FDA approval or any regulatory approval. What you finally end up when you test 10,000 compound is just one compound that has to be, that will be approved and used in human beings. Now, we know that there are different stages of this research. It could be preclinical, it could be clinical. Let us see how that works. So once again, to set the expectation, I will be talking about 3 different topics today. The first one will be the key one that we have been talking about, that is drug discovery. After which I will be talking about the application of scientific research in there. And then that is to show justice to the topic. Like, I know you guys are interested in research, and I need to tell you how the research that you are doing or intend to do will help drug discovery. And finally, I also will touch upon the career options in pharmaceutical industry, if that may help, because you already know how academic research works. Like, you do your bachelor's, then you do your master's, and then you go for your PhD. Whoever is interested in the research, whoever has that research bug bitten, they go into the PhD, and then if they further pursue research, they go into postdoc. And nobody usually talks about the industrial research where equal or more opportunities are lying. So I would like to take a few minutes to talk about the career options as well there. So the whole process, like I mentioned, it takes about an average of 10 years. It involves drug discovery, clinical trials, and then FDA review or any regulatory body that is going to review it. And then the drug will be approved and then marketed. So usually it takes a time of, anything between 8 to 10 years or even more, depending on which drug they are developing or which disease they are trying to treat. So these drug development process can be classified broadly into preclinical and clinical trials, but there is a lot more to it. In the preclinical trials, you will see that a molecule needs to be identified, and then chemical structural studies has to be done. A lot of other studies have to be done, and then it has to be tested in cell lines, in tubes, in glass plates, or in test tubes, or vials, and then in cell lines, and then it will be tested in animals as well. You know that. Then once you see any success on any drug that has been tested, if you see that, okay, you have tested few of those drugs and one is working, then you will do a formulation and then see if you can scale it up, if you can produce in a larger quantity. Once you're successful with that, in the preclinical stage, what you do is you will file for an IND, which is called Investigational New Drug Application, which means that you are telling the regulatory body. Here in US, it is FDA. In India, it's called Central Drug Standard Control Organization. So whichever, in Europe, there is a different one. There is a different one in Canada. There is one in Australia. There is one in Japan. So wherever you're doing your research, you will file an IND, which means that you are intending to test this drug, which has been showing some effect in some human beings. So if your application is accepted and if you get approval, then it goes into the second stage, that is the clinical studies. There it will be tested in human beings, and it will be going through different phases. And then if that shows any success, if that see any success, an NDA will be applied, which means it is a new drug application, which means that you are saying, I have found that this drug is safe and it has some efficacy, which means that it is working in certain indications. That is indication by indication, I mean disease situation. If it works in curing a particular disease, and if it is safe, and if it is showing efficacy, they will apply for new drug application, which is like, okay, I have this, and these results. I would like to seek approval for using this drug and marketing this drug for this particular disease. So that is the basic process that is involved. Now, in the next slide, it is a little busy slide. I'm sorry about making it a little busy, but I have it's a little bit of understanding about drug discovery process. It involves I've split into 4 of those. That is research and development, preclinical studies, clinical trials, and review and approval, and what happens after the approval. So I'm going to jump deeper into the process that I have just mentioned, and we will break this down into 4 different sections. Research and development, that usually takes 3 to 6 years. It targets the identification of a molecule, which involves understanding of a particular condition. Give me a second. Which involves the understanding of a certain condition, which means that you need to understand the disease. What is the disease? Why is the disease happening? What is going on? So you need to understand in and out of the disease so that you can find out where we can target to cure this disease. What molecule can be used? How it can be used? And once you identify a target, you screen a lot of compounds to see which can be used, which one can be used. You may come up with 10,000 compounds, and you may identify one compound which is the lead compound that you'll be using. I have an example of a signal transduction process. There is a signaling pathway. We call it signal transduction or signaling pathway, wherein one ligand, it's a simplified form of it. I have a complex form of it coming up in the later session. I mean, in the later slides. A ligand is binding a receptor, and that receptor is getting activated. That activates a compound A, then that activates a compound B, and then there is a response. So what happens in a signal transduction is that, for example, in a disease condition, this response is too much, because of which you see a disease condition. So now you are the scientist. You will think, okay, I'm getting too much of this particular response or this particular signal. And this is causing a disease. How can I control it? You can think that, okay, I can control the ligand, or I can block this ligand binding on the receptor, or I can deactivate the receptor, or I can do a process where this receptor is not activating the compound A, or the compound A is converted into B. How can I stop that? Or B is giving a response. How can I stop that? So you can be applying to see which of these stages can be blocked, and how can you block it? Which compound or which drug can be used, and how can I stop this process? So this is the basics of drug discovery and identification of a drug. So thousands of compounds are screened, and a lead compound is identified. And for this process, drug identification involves, like I said before, this process might involve a lot of information. It might involve first of all, you need to know which disease you are going to treat, and you need to know in and out of it what causes it, what are the manifestations of that, what are the symptoms of that, what is the first stage of that, what is the later stage of that, what happens if it becomes chronic, things like that. And once you know all this, then you find out a target space where you want to use a drug, and that target space should have a binding site. This drug should go and bind, and there should be a docking process. And all these processes need to be understood so that you can effectively design and use the drug. So that involves the drug identification. And then there is another thing that usually happens in the pharmaceutical industry that is called repurposing. And the drug repurposing is something like a drug has been designed and has been approved for a particular disease. Now, a scientist like you guys comes and says, okay, I think because this drug works in this manner, I think this drug can be used for another disease as well, which means that you are trying to use it for another purpose as well. And that is called drug repurposing. That happens all the time in the pharmaceutical industry. I can tell you a very, very good example in the case of COVID-19. When the COVID-19 came, even before all the vaccines were being investigated and vaccine studies were going on, people all around the world were, all the pharmaceutical companies were trying to see if their drug, whatever the drugs they have, that can be used to cure COVID-19. For example, hydroxychloroquine was repurposed to see if this can be used against COVID. So that is another process of how to deal with or come up with a new drug. Now, a lot of artificial intelligence as well as machine learning and things like that is the new thing, right? You all know that. So that is also being applied in the field of drug discovery as well. Structure, if the drug has to bind to a particular molecule for its action, for activation or inactivation, it could be anything. Maybe a compound is synthesized less, and because of that, there is a disease. You have to activate a particular pathway so that compound is synthesized in more quantities, and then you can cure that disease. So it could be any of those processes. For that to be exploited, you need to use a structure-based study, like structure-activity relationship analysis or things like that. Or a computer-based designing can be used. Maybe thousands of designs can be done, and maybe come up with one compound that could be one or two compounds that you think is effective, and then primary assays are done, and then now a lot of processes are it is automated, and high throughput mechanisms are available now. And once you identify the active compound, you test them in the mammalian system, like animal model, or in organ systems as well, and then goes into the clinical trial. And then the research continues. Okay, this drug is effective only 50%. I need to increase the efficacy to 80% so that I can cure the patient better. So what structural alterations can be done? Things like that. That can be, simulated by using, computer programming and things like that. Bioinformatics, all those things are applied. Then comes the research and development that I first mentioned was all the bench research that happens in the lab. Then comes the preclinical studies, where when 10,000 compounds are tested, about 250 compounds reaches the second stage, that is, preclinical studies. It usually takes a few years maybe, and then it involves in vitro studies as well as in vivo studies. In vitro, definitely that it means that all those tests that you do in a test tube or a petri dish or something like that, and it is outside the living system. You are trying to see if your compound is actually binding to the locus that you are supposed to, that you want it to bind, and maybe inhibiting or activating a compound or not. You can do that in vitro. Then comes the animal-based testing, wherein you use animal models, maybe a mammalian system, maybe monkeys or maybe rodents. And usually drugs are tested for toxicity, safety, and then efficacy as well. Efficacy means effectiveness. And then drugs are, drug toxicity has been tested at least in 2 mammalian species. So like in the introduction, it was told, this process is very, very laborious and time consuming and money consuming. And there are so many stipulations before even you put it into the human system, because any drug, even if it costs billions of dollars, you cannot afford to have one person die because of the drug. So always the benefit should be better than the risk or the safety issues or the toxicity. So all the steps are taken to make sure the drug is safe. Then comes the interesting part, that is the clinical trial, wherein what you do is the drugs are tested in various phases of the trials, that is phase 1, 2, and 3, and maybe sometimes in some cases phase 4 as well. So I will explain this process a little bit in detail as well. Phase 1 is when you study, after you do all the preclinical studies and then come over to once you file investigational new drug application, and if it is approved, then you get the green signal to go and test in the human being. But at the same time, you do not start testing directly into patients. What you first do is you have to make sure that these drugs are safe in humans, because you have never used this drug in human beings before. So first what you do is you have to try that in healthy volunteer individuals, very few in number, very few in number, maybe 10 or 20 or maybe 30 people. And then make sure that this drug is not causing any safety issues. Usually, dosing also will be studied. I will explain that in another slide. Then if the phase 1 trial is, usually what is tested in the phase 1 trial is not that if the drug works or not. And you cannot do that because you are not testing it in a patient. You are testing it in a healthy individual. So what is tested is safety. If this drug is toxic, if this drug is causing any complication, any organ systems are failing, something like that. Once that is done, then you go into the next phase, that is phase 2, which is done in few hundred people. And what is tested in the phase 2 is safety and efficacy. First, safety. Safety is always the priority. If even one patient dies, the clinical trial will be shut down. You should remember that it has been like 4 to 6 years already into this pipeline. And if some safety issues come up right now, you have already spent millions of dollars, and then you have to shut down the whole process, whole clinical trial. So safety has been taken with utmost care. Then once you see that this in the phase 2 it is safe, you then look at the efficacy, which means that you see if the drug is affecting in in this, but if it is effective in this disease condition or not. Now, when I say that, of course, I mean that the phase 2 is tested in patients, and a few patients are called up, and safety being the primary in the phase 2, that will be tested. Once it is successful in the phase 2, then it goes into phase 3. That is where more and more number of affected individuals or affected diseased patients, patients with the particular indication of the disease is tested. It could be a few hundreds to a few thousands of patients, depending once again on the indication. Once again, I remind you, indication is the disease. That is a certain term used in the pharmaceutical field. So more number of patients are being tested. And what is more important here is, because you already established the safety of this drug, now you're looking at the efficacy in more number of people. More the merrier, more the better. You have to see effect in more number of patients. What if you are selecting a particular set of patients and then showing efficacy and not showing a complete representation of the entire population of the disease? Then that is a problem. So you have to select, do a proper identification of the patients, which will represent all the spectrum of the disease, and test the drug in the phase 3 trial, which will have more number of patients. And a larger population, the efficacy is established in a larger population. And then you might have heard a controlled trial, like the COVID vaccine test. The COVID vaccine clinical trial was a randomized controlled trial, which means that you will give the drug to a drug as well as placebo. Placebo is something that will look like the drug, but it is not a drug. It could be a sugar solution. It could be just water or anything, but the patient will not know what he or she got. He might have gotten the drug, or he might have gotten the placebo. What will happen is, if he got the placebo, he should not show any drug effect. If he got the drug, he should show the effect of the drug. If the patient is able to know I got the drug, they might have a tendency of seeing the effect of the drug inside them, even if the drug is not actually working. That is the mentality. That is how human nature works. So that is why it is completely blinded. Controlled trial is done usually in the clinical trials, and that is what is the most efficacious method of testing a drug: randomized controlled trials. So once that is done, you move on to the next phase. But before I move on to the next stage of the further explaining about the clinical trials, I would like to bring in the collaborators, how usually the clinical trial works. Usually the clinical trials are funded by big pharmaceutical companies. It can be big or small pharmaceutical company. They act as the funding agency. Sometimes the government bodies as well do the funding, but usually it is the pharma company. But the pharma company doesn't have patients at hand to test their drug. What do? They collaborate with hospitals and academic hospitals. There are different types of hospitals, right? It can be an academic hospital where research is also done, like a medical college, or it can be done at a private college as well. So it depends. But you have to see a collaboration between a pharmaceutical company as well as a hospital, where only at the hospital you will find the patient. And this trial, when you give the drug to the patient, has to be supervised by an expert doctor. So what happens is, the pharmaceutical company collaborate with X number of doctors, X number of institutions, where they will work with X number of principal investigators, which basically are the doctors, under which there will be X number of patients enrolled into the trial. And in a controlled manner, the drug will be given to the patients, and they will be constantly monitored for safety and efficacy. And then the results will be collected. And depending on the design of the trial, it can be a few months to maybe a couple of years or more. and the data will be systematically collected. The data will be analyzed at each stage of the trial, maybe after 6 months, then after 12 months, then at the end of the trial. And at the end of the trial, you will analyze and see if the drug worked or not. So what usually is tested in the clinical trial is the dosing and the safety is tested. But before that, you need to understand how will the molecule affect the body. Just the molecule that or the drug that you are giving might be treating the disease, but it might affect other parts of the body as well. Few of those cells, few of those organ systems, things like that. And at the same time, how the body affects the molecule. At the same time, like I said, liver might be metabolizing this drug constantly. And how does the body react to the molecule as such? Like the molecule might be broken down very fast, which means that the drug is not getting any chance to work and cure the disease. Or some molecule will be metabolized very slowly, which means that there is time for the drug to work. It depends on the speed of the drug, how the enzymatic activity and things like that. And the other thing that is into consideration is, once again, is it toxic? What are the risks? And then when does it become dangerous? Like what dose becomes dangerous? Now going into that particular aspect, that is safety. Is it safe? Is it more safe or is it more dangerous? It might be causing some harm to some organ system. At the same time, is it giving some efficacy? Is it safe to use? So that is one of the major aspects that has been given consideration and being tested. Now, dose finding is also very important, which means that this usually happens in the phase 1, wherein a particular compound is taken and given in small increments, like 0.5 milligrams. Then if that is being tolerated by the individual, then you give 1 milligram. So you keep increasing to dose limiting doses. Like where do you the dose limiting toxicity levels? You increase the concentration slowly to the dose limiting toxicity levels, and say, okay, up to this particular concentration, maybe 40 milligram per individual or 40 milligram per kilogram is safe. So you keep increasing, like maybe you started at 0.5, then you gave 1 milligram, then you gave 2 milligram, then you gave 4 milligram. You slowly increase and say, okay, this is the maximum an individual can tolerate. So that is where that is how you decide the doses. And then even if the individual is tolerating 40 milligram, that might not be the advisable dose for treating the disease. It might be 20 milligram. So you need to do dose finding studies. That is usually done in the phase 1. And then comes one of the most important part, that is pharmacokinetics and pharmacodynamics. What is involved in these is how quickly it starts working, is studied. Like it is given in a healthy individual and say, okay, the patient consumes it as a tablet. How soon you see that in the bloodstream? Like I had that drug now, and within 20 minutes I see that in the blood, which means that at every 20 minutes or 30 minutes your blood is drawn and tested for the concentration of the drug, and see what is the speed, pharmacokinetics, how quickly the body can process it, how quickly the liver can break it down, which means that, okay, the drug was consumed at 10:30 and it is available in the bloodstream till 12:30, after which the concentration drastically decreases, which means that it has been metabolized and broken down. So you are effectively getting 2 hours of working of the drug only. So how can you increase the metabolic activity of the drug, slowing down the breakdown by the body, and things like that are also tested. Because even if you have a working drug, what if that is being broken down by the body so fast so that it doesn't get any chance or any time to work? So there are many research aspects into this, wherein you test to see what is a sweet spot where you can give the right amount of drug for it, so that it works for X amount of time, so that slowly the patient gets the benefit of that. And how quickly the body eliminates it is also part of it. So the whole reason I wanted to include this particular slide was because there is a lot of job opportunities in this particular area. and all the rest of the world usually outsource this particular part of the job to India. A lot of jobs that is a special reason that I wanted to mention this particular aspect, the pharmacokinetics and pharmacodynamics. Now, when you're studying biotechnology or microbiology, it doesn't mean that you cannot do this. There are ways you can get there. Like I had done MSc microbiology. I'm doing neuroscience and oncology right now. You can reach anywhere you want to if you explore the right path. I will let you know how. Now, once that is done, finally, after the third stage of the drug clinical trial, once you see that the drug is working, during all this process, like the stage one, phase 2, phase 3, you will constantly work with the regulatory body. Here in the US, it is FDA. You will keep constantly giving the data to them saying that, okay, we have reached this stage, the drug looks safe. We have reached this stage, the drugs look safe as well as efficacious. Then we have reached the phase 3. This is how the data looks. I would like to present this and submit a new drug application. That is for application for approval to market the drug for treating a particular disease. Now, they take their sweet time, like, half a year to 1 year. They can take that much time. It's called, and they will give, once you file the application, they will give a date called PDUFA date, which is the prescription date user fee act, which means that, okay, after 6 months or after 8 months, or after 10 months, on this particular day, on or before this date, we will let you know the result of your our review of your drug. So that is how the regulatory bodies work, and they do very thorough scrutiny of your data and see what you are saying is right or not. Then comes at some the phase for the review, approval, and the phase 4, wherein, like I said, the evaluation happens by the regulatory body, and then approval based on the data, and then if it is approved, the drug is manufactured and marketed. Like, about 80% of drugs that are being used in the, I think 80, don't quote me on this one, but I'm pretty sure more than 80% of the drugs are manufactured in India. So there is a lot of opportunity in that area as well. You might know, you might not know. But the problem here is that not all drugs that are applied gets approval. Very, very few number of drugs go through and gets approval. So once the drug is approved, you have the regulatory body like FDA will tell you that, okay, after the approval, you have to monitor the patients, and every 6 months or 3 months or 6 months or 12 months, you have to submit a report saying that these many patients were treated with this particular drug. What was the result? How many safety issues were there? How many adverse events were there? How many patients got treated? All those data has to be submitted. Once again, they will keep monitoring, and if they find any after the approval, if it doesn't mean that you can always sell the product. If they see any problem or any safety issues, they will tell to recall the drug, and which means that is end of the story. You cannot use that anymore. You have spent like $2 billion to manufacture that drug, and all of a sudden there is a safety issue. I will tell you why that can be a problem, because you have already tested in the phase 1, 2, and 3. It is, if you have done an experiment, I'm sure you have done experiments. If you have done it, you will be looking for different variables. That is how lab experiments are planned. There are 4 different variables. You will keep 1, 2, and 3 constant and leave the 4th one as the variable and test it in a lab experiment, and then see no change. Then you will say that, okay, first one, second one, and the 4th one I'll keep constant, and I'll change the 3rd one and see if there is a change. So that is a controlled setup, how you do experiments. So you cannot change 2 factors and see if there is a change or not. If you change 2 out of 4 and see there is a change, how do you know which one caused the change? Is it both of them? Is it one of them? So you have to test one at a time. So similarly, the research, the clinical trials are also done. What happens is the clinical trials are always done similar to the lab experiments. The clinical trials are also done in a controlled environment to produce certain set of data. But once it comes out, the drug comes out, it is given to the general population who has the disease, and what you see might be a little different or a lot different from what you saw in the clinical trial, which means that you have to follow up and see how it works. So there is a lot of things to do in the phase 4, which is the post-approval surveillance, which you are looking for safety and efficacy, continuously monitoring to see that patients who are taking this drug are still safe. Now, this is why you are looking long-term side effects, long-term toxicity, if the drug is causing cancer, gene mutations, reproductive harm, or neurological complications, things like that. That is what is looked at in the long-term monitoring. Also, in the long term, risk and benefit are also evaluated. Risk-benefit ratio is looked at. And one of the fields that is very important in this area is called health economics outcome research, HEOR. The reason I'm mentioning is that because there is a lot of job opportunities in this area. What is usually done is post-market data is collected. It is constantly analyzed, and as and when required, the changes are made in the dosing, or the FDA might need it, or for the future purposes. Or, for example, a drug was tested for a particular disease. What if that patient has another disease? So that is a new set of data. So that has to be constantly monitored. So that is where the health economic outcome research is done. So that is a good area that you can explore as well. And I don't know, since I'm not getting any response or cues from you guys, I'm not sure if you're following me or not, but I'll still continue. What my intention is to share this slide deck with you all. Whoever is interested, I will share it with the organizers. They can distribute the slides. If you think it is beneficial, you can use it for your purpose. Only for individual use, do not distribute it in a public platform. Now, once I've finished talking about the clinical research and how drugs are developed in that area, I would like to switch gears into application of scientific research, which might be most of your area of interest. So I have talked about the clinical application, the clinical research. Now I'm talking about the application in the how scientific research can be applied into this whole process of drug development. So I'm trying to switch gears here so that you know what you might know up till now in your science career, like in a bachelor's or master's, or whatever you have done until now in research, or what you intend to do in the future. So how to apply that into the drug discovery and industrial research. I will talk about the other aspects of that at the end of the talk as well. So once again, this is not to scare you. It is a very busy slide. It is a complex cartoon that shows signal transduction, and you can see that so many of those cellular processes are shown here. Like I said, it is not to scare you, but I wanted to tell you that you can see gene expression here. So there are so many protein synthesis happening here. Cell cycle is also happening here. All these processes are hand in hand, parallel, happening in parallel. And you might want to control or stop the cell division to control maybe cancerous cells, or you might want to increase the gene expression, or you might want to decrease the gene expression, down regulation or up regulation, any of those processes. So you can see that there are various stages and steps in the signal transduction or signal transduction pathway that leads to a process. So you are the expert who is going to decide, okay, this signal should be blocked over here. This is a slide I was mentioning from the previous simplified form of this. So this is one of the areas that I worked in my PhD. So this works in a maybe 20 or 30 different processes inside the cell. So you never know. You are the experts who are going to identify the process. Who is going to say that, okay, this is the pathway through which this molecule is being synthesized, and this is the pathway that we can stop at stages 1, 2, or 3. There are 3 different stages. 1 becomes 2, 2 becomes 3, 3 makes a gene expression, like 3 makes a gene transcription and translation possible. So which is the stage that you will stop? At the stage 1, stage 2, or stage 3? Or the gene expression as such, maybe the DNA polymerase is stopped. So that is up to you. Like, but once again, if you stop stage 1, the stage 2 might be involved in 2 other processes as well. So what will happen? If you stop at 1, 3 processes are shut down. What if you stop at 2? Maybe only 1 process is shut down, or maybe 2 are shut down. But when you stop at 3, maybe only 1 is shut down, or 1 is partially shut down. So that is where your key knowledge has to be utilized in identifying the disease pathway or signal transduction pathway. This is just a aerial view of how things are usually done in the pharmaceutical industry. This need not be the typical path. This is the typical pathway, but this need not be the case all the time. Now, the time has passed, and the field has evolved a whole lot. There is a lot of personalized and precision medicine coming up now. Previously, previously actually, it was the condition used to be like, one treatment fits all the people. Like if you take human population, Chinese people might be having a different genetics than Indian people. Indian people might be having a different genetics from England, people in England. And those people might be different from people in Mexico. And the same drug might work differently in these different people. So you need to understand the population. You need to understand the mechanism of action. You need to understand their genetics to see how to treat it. Previously, it used to be just one pill for all the people around the earth for this particular indication, maybe diabetes, maybe hypertension, or anything of that sort. Now, when you go closer in, get advanced into the scientific field, this is the future of medicine, where there is personalized diagnosis, personalized medication, precision medication, etc. I will tell you one very simple example, wherein, for example, 100 milligram of a compound is inhibiting synthesis of 4 different genes responsible for a disease, versus 4 different medications in different quantities inhibiting 4 different genes. I'll repeat, one drug is inhibiting synthesis of 4 different genes, shut it down completely, versus you are using 4 different drugs for inhibiting. The first drug inhibits gene 1, second one gene 2, third one gene 3, fourth one gene 4. But all these genes are contributing towards one disease. For example, it might be diabetes, it might be a certain cancer or something, but you can decide to shut down all 4 of it. Maybe in one patient, only one gene has to be shut down. That is the one that is causing the disease. Maybe in another one, there are 2 genes that are acting in that particular disease. So what you would do is you have to identify that gene instead of shutting down gene 1, 2, 3, and 4 that could be causing the disease. Or you can pinpoint one and say, okay, I do not need to shut down gene 1, 3. I mean, I can shut down 1, 3. I don't need to shut down 1, 3, and 4. I can only shut down 2 to cure this disease, which means that the side effects are less. For example, in the top part of this cartoon, you can see that there is an effect in certain population, there is no effect in certain population, and there is a lot of adverse events in certain population. And in the precision medication, what you give is one set of medication to one, another set of medication to another population, and you see a lot of effects. So that is where the science is evolving to. Now, molecular and immune profiling is also constantly done. This is done so that you can identify the correct cause, and you can identify the correct treatment as well. There is molecular profiling, which means that you identify which molecule has gone wrong, or which molecule is overexpressed, or what part of the gene has been mutated. That is the molecular profiling. You can also do immunoprofiling, wherein you do a lot of immunological experiments and then pinpoint and say, okay, this molecule is overexpressed or this molecule is underexpressed. So that is the next level as well. Then comes the use of biomarkers, wherein a lot of biomarkers are molecules that will tell you a lot about the disease condition. It can be used, okay, saying that a molecule is a lot is hyper expressed. A lot of molecule is expressed in a particular condition, which means that you can see if that molecule is the level of that molecule is very high. You can use that to diagnose a disease, and you can also use that to determine how this patient is going to respond to a particular treatment. That is like, to predict how it goes. That is the prognosis. You can also use molecular markers to say, okay, there are two molecular markers. If this molecular marker is higher, which means that we can use drug X. If this molecular marker is higher, we can use the drug Y. So you can use that for predicting what kind of treatment can be given. And you can also monitor to the same molecular marker. So for example, the molecular marker, this one is higher. And during the course of the treatment, if the molecular marker is coming lower and lower in concentration, which means that the disease is getting better. So you can also use that for monitoring the patient as well. So this is an area usually heavily used in the oncology area, wherein a marker has been showing up in a particular cancer. So you can use that to diagnose and do the prognosis as well. Now, gene therapy, I know you all are aware of gene therapy and immunotherapy as well, wherein a gene that has been, do we have time constraints or should I make it shorter? I'll take that as a no. Wherein if you identify one of the gene is mutated because of which the disease has been caused, you can use gene therapy to give a actual copy of that, deliver that into the cellular system or tissue to synthesize the corresponding protein and cure that disease. That is the gene therapy. And you can also use immunotherapy as well, wherein you can use immune cells. You can use a certain set of immune cells to cure a particular disease. I don't know if you know that cancer we can fight cancer using our own immune system. And cancer cells are very, very smart. They are very smart. They are like viruses, like they are very, very smart. They can create a tumor microenvironment. Like there is a tumor around which they can create a small environment which is not penetrable. You cannot penetrate it easily. So even if you give drugs, the drug cannot reach the tumor. Even if your immune system try to attack it, it has made a cover for itself, which means that your immune system is not able to fight it. So there is certain drug delivery mechanism that is being used in the immunotherapy, wherein this environment that is created by the tumor, which is called the microenvironment, can be targeted. And this can be targeted using antibodies or something like that, which is tagged with a drug that can target the molecule, that can target the cancer and try and cure the disease. So that is another area that is developing a whole lot, which is called the immunotherapy. And finally, there is a lot of opportunity in medical devices as well. That is another area where clinical research is a must. If you want to use devices, you might be wondering, any medical devices like a stent or anything that is used inside the body or outside the body has to be tested in human beings before it is used for human interventions or for treating diseases. So there is a lot of opportunity in that area as well. Now, once again, I would like to switch gears into a career in pharma. I would like to stress that I am touching upon this area just because I did not get a lot of guidance in my, in the time when I was going to school and colleges to, shape my career. Since I gained this knowledge, I thought I will give it away and then try and help as many people as possible. I would like to, the biggest problem that I faced was that I did not know what are the opportunities out there. If I knew different opportunities, I would say, okay, out of these 10 different positions, I like this position that fits my personality. How do I get that job? What would it take to get that job? So nobody told me that. I'm trying to, collect as much information as possible and give it out there so that more people are aware of something else than PhD and postdoc. There is a whole different and beautiful world out there that could be exploited and utilized for making up your career. So I would briefly go into that. Pharmaceutical sciences include a lot of disciplines, including medicinal chemistry, analytical chemistry, structural biology, biopharmaceutics, pharmacokinetics, like I mentioned before, pharmacology, maybe pharmacodynamics, how the drugs work inside the human body, where does it go, how long does it stay, how easily does it get destroyed, how easily does it get metabolized, how easily does it get excreted, what are the residues staying back, things like that. Those things can be studied by any upcoming science aspirant. And then once again, the toxicology, if there is any toxic effects and things like that inside the human body. So these are different disciplines. And if at all, if you think industry is your goal, when I did my PhD, industry was my goal, because after my bachelor's and master's, I worked in industry before I decided to do my PhD. So after my PhD, I wanted to always go back to industry and serve the industry. And I will tell you why. This is one of the moment of truth. You would see that, okay, by my academic research, I'm going to change this world. Yes, you might be able to change the world, but you are not having a surety of that. You might end up doing a PhD, you might produce a lot of data, you might maybe publish, then you might do a postdoc, and your research may be useful for humankind or maybe not. You never know. But in the pharmaceutical industry, what happens is, on a daily basis, on an hourly basis, I know that I'm touching life of human beings. I am working in a rare disease space, hematology, oncology, and I know that how many patients are there, how many patients need which kind of interventions, and which are the doctors treating which patient. How can I help the doctor to make him understand how this drug works, and which are the set of patients he might be able to treat. I work with the treating physician to tell him or her how our drug works, which are the patients they might be able to treat, or in an hourly basis, I'm able to touch lives of suffering patients who has, it's a rare disease. There is no treating drug as of now, and there is one drug that came out recently from our company. So I know that it is working. I know that this is helping patients. So you can literally see the effect of your work in the real life. And versus you might do a research in a laboratory. I'm not against it. I've done it for years, like maybe 15 years, and I moved on. But there is an opportunity where you can actually serve the human being and make their life a little better. Now, if you come up with that mentality, if you want to work in the industry, you can focus on a therapeutic area. This I'm talking about in the animal research. It is different in the plant research. Then you can identify the role. Like I said, I did not know what I wanted to do. I was a scientist, I could be a good scientist, but I have done enough of scientific research, so I wanted to move on. But I did not know what industry roles are available. Now you can go through different industry roles and find out what is that role, what do they do, what does it take to become that take that position, become that individual, or do that job. What qualification does it take? What are they doing on a daily basis, on an hourly basis? How can I become that person? So that is how you have to back calculate and find out how to reach there. Now, once you know where you want to go, then you improve your skills, both scientific as well as interpersonal skills. I cannot stress enough on the interpersonal skills. If you have 100% of skills, but you cannot show it or translate it, or you cannot convince people, you cannot talk to people, you are having no skill. Even if you are 100% skilled, people consider that you don't have skills because you cannot speak or you cannot communicate that. That is the end of the story. So you have to develop interpersonal skills and then network with people in the industry and say that, okay, this is this person, I know that person's name, I know what he's doing, I know where he's working, I know what his jobs are. So I would like to talk to him and say that I would like to introduce myself and say, I'm interested in your position. When I grow up, I want to be you. Can you tell more about that? You just talk, you just be confident and talk to them and network. That is what you do. And where can you contribute in the product life cycle in the pharmaceutical industry? It is endless. There is a lot of opportunity. Industry scientists, for every single stage of life cycle of the drug development, you work in collaboration with the scientist, which means that you can be a scientist in the industry, pharmaceutical industry, and then work in every stage to help the drug development. It can be a pharmaceutical postdoctoral as well. There could be a product specialist. You could be working in the manufacturing or operations. You can be, working from home, being a scientific writer. It's a lot of market in India for that. Then comes medical science liaison, which is what I am. I'm the happiest doing this particular job, which means I'm helping patients, I'm helping doctors, I'm helping the industry survive, industry get better, the treatment landscape get better. And clinical trials is an entirely different universe where the job opportunities are endless. There is all sort of job opportunities. You just need to have interest and look it up and find out what is more conducive for you. Then comes the product manager, director positions available in the industry, which means that they are the one who manages the drug once it is in the market out there. Then comes the regulatory affairs, who works constantly with FDA and people like that to make sure that every step of the research is compliant with the regulation. And then field application scientist. You must be a scientist in the field who is supporting the product, or a data scientist. You know that data is everything now. It's all upon data. Drug discovery or drug design, everything depends on data. Now, even a lot of data is being generated from patients as well to see how that can help the treatment landscape as well. You pick and choose one set of patients with X, Y, and Z characteristics and say that, okay, this drug is going to work on this one. That is where the world is going to. Marketing is also a different option, if you prefer to do that. So before this is going to be the last slide, and before I finish, I would like to just set some fire up here and then see if people are committed or not. And just wanted to make yourself ask, are you just doing it for studying purpose? Are you just there to get the degree, or are you just the science nerd within the four walls of the lab or something? Or do you actually want to give it back to the society? The reason I took 2 or 3 days to prepare this entire presentation. Why am I doing this? Because I'm giving it back to the society. Because the education, lot of you, even if you're paying your fees for your education, a lot of it comes free without your knowledge. A lot of it is taken care by the society that you're living in. A lot of it is taken care by the government as well. So you have the obligation to give it back. That is the reason I'm doing it. So you think for yourself and see if you want to be responsible for yourself as well as your surrounding, and what can you do with the hard-earned knowledge? You are sitting right here and listening to me. Why are you doing it? You are gaining knowledge. If at all you are doing it, you are gaining knowledge. You are doing a bachelor's, you are doing a master's. Why are you doing it? Why? What is it going to do for you and the people around you? Are you going to use it for making the human being's life better? If not, what is the purpose of science? Ask that question when you do every day. When you do science, ask that question. You might be sitting at home and doing your learning because of COVID, but think out of the box and find out how you can contribute and see what does it take for me to give it back to the society. And then one final thing is that the moment you start becoming comfortable in your situation, like you are sitting at home and learning, I'm comfortable here, you are just going to be at home and not being comfortable there. Just be comfortable. Being uncomfortable is a great quote by a great person, and gain intellectual and financial independence. That should be your ultimate goal. With that, I would love to conclude this talk and then wish everybody the best, and then take any questions that you have.
Yes, participants, if you have any queries, you can post it in the chat box, or you can unmute yourself and straight away put forward. So before
please talk to me. Please talk to me. Anybody, please talk to me. Do not be reserved. Don't be afraid. Just talk to me. I'm just like a person like you. I came from the same place that you all are.
So we have a question that has come up in the chat box. Shall I read it out?
Absolutely.
Yeah, it is from Sheetal Pradeep. What were the challenges that you faced during, just a second. What were the challenges that you faced during your tenure in a foreign country?
Oh, the biggest one is the cultural difference. Definitely, that is a cultural difference. Initially, I just wanted to run away from here, go back to India. But you have come here for a particular purpose. I wanted to have a better, quality of education. So that is the reason I took all my effort. It takes a whole lot to do what, to do that. So if I come here, if I have cultural problems, there is all sort of difference. If you cannot survive you might not be able to survive other things as well. So cultural difference is the biggest problem that I faced. Then financial issues. Definitely there was financial issues. I came by myself. I did not depend on anybody. So that was another big issue. Family back home. It's not just the culture. You are cutting off from the family. I have only one relative in this country when I came here, and I still have only one relative other than my family right now. So those are the challenges that you face. Plus the expenses, like the expense is very, very, very high. So what I had to, you might ask, why did I go to this particular university? Because I found scholarship there, or because I found a job in the campus, which will pay for half of my tuition fees and half of my living expenses. So those are the kind of challenges that I had to take up and live like that, like a bare minimum life for 6 to 7 years to complete the PhD. So, but it is what it takes to get a higher education. So be it.
So before we move on to further questions, we have our principal joined here, Dr. Lalitha Sia. So thank you, ma'am, for joining. So let me invite Dr. Lalitha Sia to say a few words.
Absolutely. I'd love to hear from her.
Hari Om.
Yes,
I
ma'am.
couldn't join because of the connection was not there here. Okay. The guest of the day, Dr. Sanil, and my dear faculty members and students. Today, we have organized an international edition of the Chinarts webinar series, and we all have listened to the topic and how the drugs are developed and how the scientific research takes a role in biopharmaceutical application. I'm happy that the Department of Biotechnology has chosen such a relevant topic in today's webinar. I hope the students got ample informations about the recent developments in this area. I wish all the best to all who have attended this webinar. Thank you.
Thank you, Dr. Lalita.
Thank you,
Thank you very much for your kind words.
Shall we move on to the questions?
Absolutely.
Yeah. So next question, we have it from Sauparnika. She's asking, what are the chances, according to you, that there would be a foolproof drug against COVID-19, and how long may it take?
Very relevant and very, very controversial question. Whatever answer I'm going to give, please do not quote me on that. But the chances of, I am a microbiologist, okay? I'm a hardcore microbiologist. If you ask me microbiology, if you ask me any question in microbiology, if you ask me to do something in microbiology, if you ask me to streak a plate, if you ask me to culture any bacteria, I'll be the happiest person. I have microscopes at home. I'm hardcore into microbiology. But the reason I'm saying is that this is a microbiology topic. And if you know about viruses, this is my take, okay? This is just a personal opinion from the knowledge that I gained. Viruses are obligate intracellular parasites that has a constant capability of mutating. And you have seen that with various variants of this virus, even in India. Now we are dealing with the Delta variant, right? So the unfortunate part is that this virus or some viruses doesn't mutate that fast or that frequently, but some viruses do. One very good common example is the common cold rhinovirus. Have you ever seen a treatment for common cold? You have not. Have you ever got common cold? Is there anybody here who did not get common cold? There is nobody. Everybody gets common cold. Why is there no treatment? Because this virus that causes common cold keeps mutating. I get common cold because of a virus and I pass it on to somebody else. By the time I'm passing it on to the next person, it might have mutated. It is very, very fast. But fortunately, it doesn't cause so much of a problem. Nobody dies of common cold unless you are completely immunocompromised. Nobody dies of common cold. You are not getting a influenza or something because of common cold or something. That happens only in case of a completely immunocompromised situation. But my point is that similarly, COVID-19 vaccine is also a very potent in mutation. And that is why you are having different variants. You had South African variant, you had English variant, you have Delta variant that is causing problems right now in US as well. So that being said, the question is that can you produce a drug? You theoretically can produce a drug, but will it be possible? You will have to include drug that will kill all the variants. Even then, the virus might have a possibility of overcoming that, coming up with a new mutation and saying that, hey, I'm out here once again. So possibility of getting a completely efficacious, effective drug is under question. I wouldn't, I wouldn't consider that to be an easy task. So the second part of the question is how soon? So that answers it, nowhere soon. So that being said, my personal opinion is that this disease or this virus is here to stay. It will stay.
The next question, it is from Swati Sujit. What are the challenges faced during the drug development process and how do we calculate the long-term effect of the drug?
I think I explained that in the talk.
Yeah.
But still, the long-term challenge is the safety and efficacy. It is every single stage. Like if it takes 10 years, our drug got approval after 11 years. If it takes 10 years to approve, it goes through like 20 different stages. Every single stage has its own challenges. Like I said, you might be starting off with maybe 10,000 compounds. And maybe you are getting out with 250 compounds in the preclinical stage. You know how difficult it is to do a testing inside a cell line. You take maybe HeLa cells and culture it on a plate, test that on those cells, and say, hey, my experiment did not work. Did it cost you money? It costed you money. Did it cost you time? It costed you time. So you are part of that research as well. Maybe your professor is part of that. And so you are doing that work for that professor and you are getting that particular project done into your master's or your PhD. Did it cost you time? Yes, it did. Did it cost you money? Your lab costed. So money is a big challenge. Regulatory bodies are a big challenge. The money I said for on an average, it costs $2 billion to get one drug approved. That is why drugs are very costly. It has and for example, one company is testing 10 different drugs. They might get approval for 2 drugs. How much they have spent? $20 billion, and they have to recover that from 2 drugs now. So that is why drugs become expensive. But can you say that it is costing a lot of money? I don't want any drugs. You can't say that because you need drugs. You need medication. That is how human beings have become successful in surviving this much. Because if you decide, I don't want vaccination, what will happen? So many people died because they did not get vaccine, right? Smallpox killed so many people. When we discovered drugs against smallpox or polio, we started surviving more and more. So mostly it is the money challenge. And for example, let me take one part of that 4 different stages, like the preclinical, then the lab stage, then the clinical research, and then came the post-approval stage for this one. So just consider the phase of clinical trial. If your drug is in the phase 2, you have to recruit, for example, 300 patients. Finding that patient population is very, very challenging. I work in a rare disease condition, which definitely means that the number of patients in that field is very, very less, right? So if I have to do a clinical trial in that particular area, I have a big challenge of finding those patients, because there can be 15,000 patients, but maybe only 100 people are willing to participate in a trial. So you have to make sure that, okay, why should I participate in a trial? So that I might not get benefit, I might get benefit, I might not get benefit. But at the same time, in the future, anybody who is getting the disease is definitely going to get benefit, right? Because at least I generated some data. That is how human beings develop. So that is a big challenge. Finally, when it comes to approval, it is a big, big challenge. You have to convince the FDA or a regulatory body saying that, hey, this is how it works. So they will come back and say, no, we don't think it works. So you have to sit there and convince them. So that is how it works.
Thank you, sir. Next question we have from Shreya. She want to know, as a biotech students, what are the additional skills needed to get a job in the pharmaceutical industry?
Very good question. Can you see right here?
Yeah.
This one right here. This is the best. And if you know how to deal with people, you can do this. Okay. If you have interpersonal skills, you can talk to people. You have knowledge, but you cannot communicate with people. What would you do? How will you convince people? I am ready to fund your research, but you cannot effectively communicate and say that, hey, I have a method by which you can cure this. If you are not good at talking to people, if you are not good at dealing with people, if you are not good at convincing people, you are absolutely good for nothing. Sorry to say that. So that is how the world works. And once you have that, you have to be able to. This is usually lacking. If you take 2 different cases, engineering students and life science students, who talks more? Engineering students talk more. You take economics students, you take physics students, they will talk more. And biology students will always sit in a corner. Why? That should not happen. You have to develop the interpersonal skills. I have seen this because I have come through the same path. Usually they lag behind. That is not because they can't. They are just doing it. So you have to have, other than the scientific knowledge, you have to be enthusiastic. You have to go find out what you want to do, back calculate what it takes, and you have to be able to translate that into your skills. You have to show, you may have developed your skills, but you have to show the skills out there. You have to be smart enough to put it in papers in your resume. You have to be smart enough to put that into your LinkedIn profile. So that those are interpersonal skills. If you can talk in front of me and convince me in 20 minutes, that is great. If you can't, what is the whole point, even if you have all the skills or knowledge? So that is what it takes.
Okay. Next question we have from Varsha. IPR hinders the production of certain drugs or using certain methods by other company. This results in monology of the drug. How long does IPR provide benefits to the company?
It depends. It depends on the disease condition. It depends on the drug. It depends on how many similar drugs are available. It depends on where the drug is developed. The same doesn't apply in different countries. The same drug that has property rights in US may not have that in India. I recently learned from a very, very good specialist in hematology. He is from Kerala, Trivandrum. I happened to talk to him. He said that the same drug, which cannot be marketed by any other company for another 15 years here, can easily be manufactured in India because those rules or the patent rules doesn't apply in certain countries. It just doesn't, because the drug here costs about $150,000 per year, and nobody in India will pay that much money, right? So it depends on the boundaries of the countries and regulations of individual countries. I unfortunately do not have the idea about how things work in India, but I know that it depends on the contract, your molecule, the disease condition, how much of requirement is there. For example, you have the right of the drug, but you have a large majority of the population to treat, and you have the monopoly of that. And you get to decide everything. At that time, the government might disagree and say, no, we are not going to honor your IPR rights anymore. We are going to give authority for other companies to make it, because it might be a life-saving drug. So you know what I mean? So it depends on the indication. It depends on the requirement as well.
Okay. Next question is from Fatima Azza. Now, what if the disease is a rare one and where we don't get much patients to experiment on? What will that make it less efficient?
Beautiful question. That is a pinpoint question for me, I know, because I've been mentioning about the rare diseases here a lot. Yes and no. Once again, for example, let me take the condition that I have been dealing with, like our company has been dealing with. It's a condition where we have only less than 15,000 patient population in the United States, and our trial was done only in the United States, which means that the number of patients are limited, right? So our drug was tested in a particular trial, like phase 2A or phase 2B trial, where we only tested in less than 100 patients, because that is all we could. And so the question is, can you say that I did not get enough patients for a phase 2 trial? We need 300 patients per the books. Like the book rule says that 300 patients should be there. But as a whole, active patients are only 500, out of which 250 are not eligible for the trial, because the trial comes with so many intricacies, which include inclusion and exclusion criteria. That is why, because of those inclusion and exclusion criteria I mentioned, trials are like experiments conducted in a petri dish, in a controlled environment. So because, for example, a patient comes with a lot of liver conditions. His or her liver is not working properly, which means that his or her liver enzymes are high or low. But liver has nothing to do with this particular disease that I'm trying. But liver is the enzyme, the liver is the organ that is going to metabolize the drug. If the liver function is less, the drug will remain in the body for longer. If the liver function is high, the drug will get metabolized faster. Even though this patient has the disease, I cannot use this patient because the data that I generate from this patient is not reliable, because there is another variable that is affecting the efficacy of the drug. So you sit across a table with the regulatory body and say, this is reasonable in this indication, in this rare disease. You can only find this many patients. And there are diseases wherein I think when it is less than 1 in 200,000 patients or something, that is when it is called rare disease or something. There is ultra rare disease as well. You know, there was a case of some neurological disorder in Kannur where there was crowdfunding, and that was very ultra rare condition, and and the drug was shipped from US and things like that. So if you want to do a clinical trial in that indication, can you do a clinical trial? You cannot do a clinical trial, right? So it once again depends on the indication and the prevalence of the disease. So you cannot say it is biased, or it is, once again, you have to first look at the total number of patients that you have, and then say, this is the number of patients that the entire population has the disease, out of which only 20% are eligible for clinical trial participation. And you still, in that 20%, if that 20% is 200 people, those 200 people might not, in that 100 people might not be willing to participate. But because I can decide saying that, oh, I don't want to have a trial drug which is not approved into going into my body. So maybe only 100 people are finally eligible and willing to participate. Then you have to conduct in that 100 people. But at the same time, you have to say that in this, that is where the statistics comes in. Statistically, you have to have the power. It's called the power of the test. You have to determine the power that you will generate to say that this drug is efficacious or not. And you, with that calculation, with that proposal, you have to approach the regulatory body and say, this is the drug, this is the indication, this is the drug, this is the mechanism of action. And if we find that out of these 100 patients, 40 patients are getting better, 50%, then we can consider the drug is working. Then the FDA or the regulatory body will say, no, we would consider that 45 patients, we should have 60% of efficacy. Then we will consider. Then you will debate between them. And finally, they will say, okay, 45 patients should get better, 55%. Then we can come to an agreement. So that is how it works. It is completely a negotiation. Then finally, when you decide that, looking at the feasibility, then you conduct the trial. So it is an agreement.
Okay, we have few more questions. Next one is from Anantyaya. Same medicine and same dose are available in different prices. Does costlier one have any benefit over the cheaper one?
Very subjective question. I cannot comment on that because it depends on the molecule to molecule. But if it is a generic drug, it basically should not. It basically should not. It might be a variation of maybe 2% or 5%, but I cannot comment on that. Do not quote me on that. It depends on drug to drug difference as well as company to company who is producing the generic drug. But hypothetically, by protocol, it should not, because a generic drug, they should follow the same formula if it is a generic drug.
Next question from Aparna Harikrishnan. Can you describe about the last project you have led and what was its outcome?
Oh man, that's a big question. Research-wise or pharmaceutical industry-wise, I need to know that. Let me tell you about. Let me tell you about the current work itself. So because of this particular job title, like medical science liaison, this can be done by different qualification, people with different qualification. It can be a PharmD, it can be a doctor, it can be a PhD doctor. Various people comes with various skill set. So fortunately, we have a group of different qualifications. So we come with different skill sets and then contribute equally. So in the rare disease that I work, it is a multifactorial disease wherein different parts of the body can be affected in different ways, and different patients respond differently to the disease as well as the drug. And because different organs are affected, there is no biomarkers available. That is one of the reasons I mentioned about the biomarker. How important is biomarkers? For example, if what usually happens in the case of cancer is that you may not diagnose it in the right time, or even if it is there, you might not be able to diagnose it because there is no diagnostic tool for that. That is where a biomarker comes in play, which means that a biological molecule is saying that, okay, you have a malfunction inside the body. So that is where the biomarker is. So one of the projects that I undertook was to identify and see if we can collect the blood samples from all the patients that we analyzed and see if we can come up with a biomarker panel. Like in the sense, I'll say, okay, we can test biomarker 1, 2, 3, 4, and 5 and say, we can develop an algorithm saying that if this biomarker is high, this patient is not going to respond. If this biomarker is less, this patient is going to respond. If this biomarker is high, this particular organ is affected more. But there is a ton of challenges. It involves hundreds and hundred thousands of dollars. It involves many parties like my company plus academic institutions, many doctors. So coordinating all these becomes, coming up with a convincing idea, first of all, becomes a big problem. Collecting a lot of background information for that is very, very important. Like all the literature you read, all the information that you collect from doctors when you talk to the doctors. So that you collect, you assimilate everything, make a proposal and convince the people saying that, hey, this is something worth our time. This is worth doing a project. So let's take it up. It's still being worked upon. So comes with a lot of challenge, but if you do not once again have the people's qualities, and if you do not know how to talk to people, how will you collect the information from doctors? How will you collect information from others? How will you convince people? How will you make a project happen? So those are the challenges. I hope I answered the question.
Okay, next question we have from Malavika M. How reliable is artificial intelligence in drug discovery?
You guys are always asking subjective questions, very, very subjective, but it is getting reliable, more and more reliable, because more data is being collected, more data is being analyzed, more data is making sense now. So even if the artificial intelligence is less reliable, you have the second stage of option. You come up with, for example, if you manually test 10,000 molecules to come up with one molecule, imagine how much time it takes. It might take you 5 years. If you do artificial intelligence and then come up with a molecule in 1 year, maybe you come up with 5 molecules in 1 year, you still have 4 years left. You can test those 5 and say if that 5 work or not. You still can test another 5 in another 5 more in 1 more year, which means that you are testing 10. What are the odds or what are the chances that you will hit success using, coming up with more number of molecules in lesser time? You have more chances. So it's getting more efficient. It's getting more, it is showing more success now.
Okay. Next one, we have it from Nilina. How can we predict or discover active site location in proteins?
Predictive analysis, molecular modeling, bioinformatics, 3D structures, 4D structures, it's all model. You can take and analyze the crystal structure of the molecules, right? So if you want to say, for example, a ligand is binding a receptor, and you want to make sure that you need to have you are synthesizing an inhibitor of that process, which means that you create a compound that binds to the receptor more actively, more forcefully, and more strongly on the receptor, so that the actual ligand cannot bind on it. So how do you do that? You need to know the structure of the ligand. You need to know the structure of the receptor. You crystallize that and analyze. Now the computer is helping you do that so easily. You can analyze the structure. The camera can go all around and find out, or you can use laser techniques and things like that, bounce off some lasers out of the structure, and then analyze it, and then find out the structure of it. Now, from the structure, you can find the active site where it is binding, and possibly like hemoglobin structure or something. That carbon dioxide and carbon monoxide, if you take those, or oxygen and carbon monoxide, if you take those two, carbon monoxide binds harder onto hemoglobin than oxygen because of the molecular affinity, right? So you can do the molecular analysis and find out the affinity. So that is where the chemistry and physics works together. And that is why carbon monoxide is toxic. Do you know that? Oxygen can bind to hemoglobin. At the same time, carbon monoxide can bind to hemoglobin in the same place. but which binding is stronger? Carbon monoxide binding to the hemoglobin is stronger. Once it binds, it will not leave. So your blood cannot carry oxygen. That is why you die. So I'm just giving an example. So that is how the predictive modeling and things like that works.
Okay, next question is again from Nilina. She want to know, how can I get into clinical research field after my master's?
Well, wonderful question. We can talk offline. I can help you with more information, but what you need to do is read, if you're really interested, if you can find, because of this era, like the computer era, and if you look up, if you Google anything, there is no difference from what I find and you find. The only difference is the method used, right? So if you are keen and interested, you can find information on anything. Like anybody sitting here, whoever finds that information, anybody sitting in Kerala or Kannur, they can find the same information. So it takes a lot of research to find out what are the positions available, which is the right one for you. But what would help is knowing what happens in clinical research. And there are so many courses, like in diplomas as well as bachelor's and master courses, that will prepare you into clinical research. If you need more information, I can provide you guidance. That is something else that I wanted to offer. Any students in this crowd who needs more information on any of those topics that I talked, I'm willing to help in any magnitude.
Thank you. Next, it's from Varsha. What aspect of bioinformatics are required in drug designing and running simulations?
I am sorry, I do not know. I have zero experience on that. I might have taken a bioinformatics course just for the sake of the degree. I do not have any further information on that. If I try to help answer that question, I will be lying. So I will not answer that question.
Okay. Next one from Safida. What are the challenges for a drug development team?
Drug development, I think that's a similar question that we already answered, right? Like the drug development, every single stage of the drug development comes with a challenge. So it could be money, it could be bottleneck where the drug is not efficacious. You need 80% of efficacy, but it is only working 50%, or the drug as such is a lot toxic, which means that more toxicity than the actual drug working. So that could be a big problem. The money is a big problem. The time duration, like, 10 years is what it takes. Imagine one company trying to produce that drug for 10 years, and you are working for that company from first year to maybe 3 years, and you are leaving and going, another person coming and join. He or she has to pick it up from there. So that long process itself is a big challenge. You know what I mean? Plus, at the end, you have to show the data that is convincing. So that is altogether a big challenge.
Okay, next one we have is from Shantini. What kind of drug development is on demand?
Oncology? I expected at least one student to ask that question. It is not fair to do that bias, but oncology is the field where maximum amount of research is happening. If at all anybody is willing to go into the industry, I would recommend, picking up, interest in oncology is going to give you a lot of opportunity. But oncology is the most challenging field as well at the same time. Most challenging, because same cancer in two different individuals will be different. Same cancer, it is called the same cancer, but two of those patients will respond differently. They will have different characteristics. Everything will be different. So it becomes a challenge in itself. But the research opportunity is endless. It is endless, and there is a lot to do. Because we haven't cured cancer, it is very hard to deal with. So the highest demand is in that area. Maybe then comes infectious disease, and then things like that, neurology.
Okay. The next one, it is from Abhishna. What are the different exams to crack to attain our aim for abroad jobs?
No job can give you, no exams can give you jobs in abroad. It is close to impossible, because if you think about the US, I can say it is impossible. You have to come here and study to get a job, unless you are a computer professional and a consultancy or a company is hiring you directly from India. That is a different story. But in life sciences, it is not possible to get a job directly. You have to come here, get a degree, and based on that degree, you can get jobs. In other countries, I don't know. I really don't, because I don't have that information. There could be maybe in Singapore or something like that, but research background is highly recommended. Like, industry facing research background, not the academic based, because there is a lot of difference between industry research and academic research, because academic research is always focused on getting more funds and getting that project rolling, and in the interest of the principal investigator or the scientist. Whereas in the industry, it is always money oriented, or therapeutic area oriented, which once again is driven by money. So it is based on different interests. The industry research is very fast paced, and all of a sudden it can stop, and you might be put on a different project. Unlike academic projects, it can be going on for 5 to 10 years in academic, but the same project can be only 2 years or 1 year long in industry. So your taste should be different to develop into the industry, yourself into the industry.
Okay, I guess that is all the questions that are so far popped up. So I thank you, sir, for patiently answering and settling all the queries. Now, let me invite Ms. Jisha M., Assistant Professor, to propose the vote of thanks. Over to you,
Thank
Jisha.
you.
Thank you. Hari Om, a warm greeting to all. May I, on behalf of the entire fraternity of Chinarts and Science College for Women, express my profound gratitude to Dr. Sanil Sansar for making it convenient to be with us today to deliver the fruitful, comprehensive, and career-oriented talk on the topic, How Drugs Are Developed, Biopharmaceutical Application of Scientific Research. I express my thanks to Principal Dr. C.R. Lalitha for her continued guidance and support in all endeavor. I place on record my sincere thanks to Naina Sahadevan, HOD, for introducing the resource person to us, and to Ms. Divya Padmanabhan for hosting this webinar. My thanks to all faculty of PG Department of Biotechnology for their teamwork and support for this venture. Special thanks to Ms. Anita Haridas, HOD, Department of Computer Application, for technical support and other help provided as IQAC coordinator. I extend my thanks to other delegates who have joined us and the participants who have made this event a great success. Thank you.
Okay, thank you, Jisha.
I would like to extend my thanks as well for this opportunity presented to me. And it has been a privilege. I should tell you one small story. I tried to get into Chinmaya Mission School when I went to school. I failed. I did not get an admission. I should tell you all that. So you guys are all more eligible than me and more fortunate than me. I should tell you that. And then keep up the interest in the science. Make sure you do that sincerely, and then contribute to the rest of the world. Your knowledge will help the rest of the world. Even if it helps one single human being in bettering their life, that should be the purpose of science. And I once again thank the entire institution, Chinmaya Mission, as the college as such, and the Department of Biotechnology for conducting this the talk, as well as giving this opportunity to me. Sincere thanks again, and please reach out to me if you need additional information anytime, or if further talks are required in a particular area of interest. I will extend my offer again to students, whoever require additional guidance. If at all I can be of any help, please reach out to me. Thank you once again. Have a good day.
Thank you, sir. We'll definitely get back to you.
Thank you.
So participants, the attendance and feedback form is already posted. Please note it will be active only for 30 minutes. Kindly fill up the details. And we'll wind up on behalf of our department, Chinmay Arts and Science College for Women. I would give my regards and appreciation to all the participants, resource person, a principal, and all the other delegates who joined us and made this event a reality. Thank you. Thank you one and all. We may now leave the session.